All right, so we left off with these events happening at the neuromuscular junction for our muscle contraction. Remember that the action potential had traveled down the axon of the motor neuron. It had opened these voltage-gated calcium channels, which caused acetylcholine to be released from the cell into the synaptic cleft. And acetylcholine bound to receptors on the muscle cell membrane. Now, these receptors are what we call receptor channels, and when acetylcholine binds to those receptors, it allows sodium and potassium to flow through the channels, and that depolarizes the muscle cell membrane in a very similar way to what we saw with the neuronal cell membrane when we talked about the nervous system. All right? The difference, of course, is that these are chemically gated channels because acetylcholine is binding to the receptor, and that allows sodium and potassium to flow through the membrane. It's also, even though I guess I drawn it, or I drew it in a way that wasn't really well represented, sodium and potassium are actually passing through the receptors because those receptors are also channels. All right? So, similar idea, identical outcome, I guess, depolarization, but it looks a little bit different than what we see with the neuronal action potential. All right? But at any rate, we have now depolarized the muscle cell, and we want to look at what happens next to get us to a muscle contraction. Right? So these were the events at the neuromuscular junction. Now we want to look at the second concept, which is excitation, contraction, coupling. Let's go ahead and do that. So step number 2. 2 is excitation, contraction, coupling. Okay. So, if we were to draw our muscle cell again, here's our muscle cell that we just depolarized. We're going to draw our motor neuron in here as well. All right, there's our motor neuron. So up here is where we saw the action potential coming down. Everything that we just looked at on the previous slide is what I've shown right here, right? So this is our neuro junction. Okay, and this is the space or place where we depolarize the muscle cell. All right, and if you remember back to neurons, we talked about how that depolarization would spread outward from that point, right? So it's similar to a graded potential. We have that depolarization spreading outwards. So those ripples, if you will, are now spreading outward from that point of depolarization. So the membrane is becoming depolarized here, it's becoming depolarized here. And what we want to ultimately affect is that organelle that we talked about earlier on in this unit called the Sarcoplasmic Reticulum. Okay, I'm just going to abbreviate it from now on as the SR. So we have the Sarcoplasmic Reticulum. Now, the sarcoplasmic reticulum is a modified endoplasmic reticulum, and it lies underneath the sarcolemma. All right, so we have our sarcolemma here. If we were to maybe somehow able to come in and peel that sarcolemma back, what we would find underneath it, kind of laying on top of those myofibrils, is we would find the sarcoplasmic reticulum. Right? There might be a couple of them in a muscle cell. It looks something like this, right? I think the artist of your textbook makes it look like a piece of sliced Swiss cheese laying underneath the sarcolemma, and that's kind of what we have. And ultimately, we want to affect this sarcoplasmic reticulum, all right? So The cell membrane has invaginations in it called T-tubules that allow that depolarization to spread and affect this entire sarcoplasmic reticulum. So let's go ahead and see what is happening when that depolarization spreads to the SR, to the sarcoplasmic reticulum. So let's take this. What we'll do here is we'll come down here and we will blow up this section of the sarcoplasmic reticulum. All right, so there's our sarcoplasmic reticulum that we just sort of are blowing up there to take a look at. On the membrane of the sarcoplasmic reticulum are channels called Ryanodine receptor channels. These Ryanodine receptor channels are abbreviated typically as RYR channels. Okay, and what happens is when the depolarization spreads to the sarcoplasmic reticulum, it causes those channels to open. So we have a situation that looks something like this. This would be a closed channel. This would be an open channel, right? The sarcoplasmic reticulum is a storage tank, essentially, for calcium. So inside of the sarcoplasmic reticulum, we find a huge amount of calcium. Okay, so this is calcium. And when this channel opens, when the sarcoplasmic reticulum is depolarized, what happens is that it allows calcium to move out into the sarcoplasm of the cell. Okay? Okay? It diffuses into the sarcoplasm. So what we're doing is we're essentially opening the valve on these storage tanks for calcium, and calcium is then going to move out. All right? Keep in mind that this is a different pool of calcium than the one that we encountered earlier, right? So we have a couple of different pools of calcium. When we talked about the events happening at the neuromuscular junction, we were talking about a pool of extracellular calcium that moves into the axon that allows for that exocytosis of these vesicles with acetylcholine. Here, we're specifically talking about an intracellular pool of calcium that is going to diffuse into the sarcoplasm of the muscle cell once depolarization occurs. So we're talking about calcium in two places. However, we're talking about two separate pools of calcium. Okay? And this calcium is necessary for the muscle to contract. Right? So this whole idea of excitation contraction coupling is really relatively simple. What we're doing is we're depolarizing the muscle, and that is allowing us to release calcium into the sarcoplasm of the muscle cell. So why is calcium so necessary for the muscle to contract? Well, that's going to lead us to our third phase here, which is the sliding filament theory. Okay. The sliding filament theory. And now we have to look specifically at those actin and myosin filaments that we discussed a little bit earlier, those thick and thin filaments. If you remember, our actin looks like this, right? It's these little individual proteins that make up this strand. It looks something like this. And it looks something like that, right? They're kind of wound around one another. Okay, and then our thick filaments are made up of this protein myosin, right? There's some overlap there. So here's our myosin that kind of looks something like this. And remember that each one of these guys has two of these little heads that come up. So on an individual thick filament, you'd have many of those myosin heads that come up, right? So each one of these subunits that make up this thin filament, every single one of them, kind of about where I'm placing these dots, has a myosin binding site, right? So this myosin head has the ability to sort of reach up and bind to this actin. Okay, these are myosin binding sites. These little these little purple dots. Ignore that one. So we have these myosin binding sites. Okay, on every single one of these guys. So these myosin heads have the ability to come up and bind to actin. And when they do that, when they come up and bind to actin, they create something that we call a cross bridge. Okay, they create something that we call a cross bridge because they're essentially creating a bridge between the thick filament and the thin filament. All right? Now, they have the ability to do this, but in a relaxed muscle cell, one that has not been stimulated to contract, they're actually prevented from doing that. And they're prevented by a couple of proteins that we need to talk about. Remember we talked about these accessory proteins. We talked about the protein troponin. And we talked about the protein tropomyosin. Okay. Specifically, troponin is a calcium binding protein. And tropomyosin has a slightly different job that we're going to take a look at right now. Okay, I'm going to leave troponin here as orange, and I'm going to use green to sort of illustrate tropomyosin. Okay, what tropomyosin does is it winds around this thin filament, kind of like this. And as it winds around this thin filament, it interferes with those myosin binding sites. In other words, it blocks those myosin binding sites. So what tropomyosin does is it prevents those myosin heads from coming up here and preventing or forming a cross bridge. All right? And then we also have tropomyosin that is bound, or I'm sorry, troponin that's bound to tropomyosin. So we have troponin that's sort of in here like this as well, and I know that that isn't coming through maybe as clearly as it could be, but we have troponin kind of like this. Okay, so tropomyosin is blocking the myosin binding sites. I'll write that down here really quickly. Okay, so it blocks myosin binding sites on actin, right? Tropomyosin blocks those myosin binding sites on actin, and troponin is a calcium binding protein that's also found in this kind of protein complex that we have here. All right. So remember that what we've done now is we've stimulated this muscle cell to contract. We had the events at the neuromuscular junction, we had that excitation contraction coupling, and the result of that was that we released calcium into the sarcoplasm. So now calcium is starting to float around in this space. And what calcium is going to do is it's going to start to bind to troponin. Remember, troponin is a calcium binding protein, right? So calcium is going to start to bind to troponin, right? So that's going to be number 1. Okay, calcium binds to troponin. Now, when calcium binds to troponin, what it does is it causes a change in the shape of the protein. And because troponin is bound to tropomyosin, it also causes a change in the shape of tropomyosin. In other words, we see a shift in the shape and the position of these proteins. And what happens is that troponin is going to be moved out of the way of the myosin binding sites. So, we actually physically are going to be moving tropomyosin out of the way. Okay. This is number 2. We have a shape shift that moves tropomyosin. Okay, we have a shape shift that moves tropomyosin. Okay, what that is going to do is it's going to allow myosin heads to come up and start to bind with those binding sites on actin. And that's going to be number 3. Number 3 over here, we're going to say that myosin can now bind with actin. Okay? And that is going to start the sliding filament theory. Okay? So I think from this, what we can do is we can see how troponin and tropomyosin really work to regulate muscle contraction, right? Remember in the last lecture, I referred to these guys as regulatory proteins, and we can see here how they are going to prevent the interaction between myosin and actin, and that would prevent muscle contraction from occurring, unless calcium is present. If calcium is present, we see that tropomyosin is literally going to be moved out of the way so that myosin and actin can bind with one another. All right? Okay, so now that we understand that, let's go ahead and take a look at what happens next.